Thanks so much for joining us for our DeFord lecture series. My name is Elizabeth Katlos, and I'm a faculty member in the Department of Geological Sciences at the University of Texas at Austin. We're part of the Jackson School of Geosciences, and the DeFord lecture series is our departmental seminar series. It's been a requirement and tradition for all graduate students since the late 1940s. The lecture series is named after Professor Ronald DeFord, who joined the university as a professor in 1948 with the purpose of enhancing the quality of the graduate program in the department. Our lecture series is supported by a range of endowments, and today's speaker is the Fred L. and Francis J. Oliver Lecturer in Texas Hydrology and Water Resources. The lectureship was created in 1985, and the speaker will be our 36th Oliver Lecturer. Fred and Francis Oliver created this lectureship because they believe the study of water resources in Texas is as important as the study of oil and gas. They established the lectureship with the goal of providing better education concerning water problems being faced by Texas and the solution to those problems. Fred received his Bachelor of Science in Geology from the University of Texas in 1951. He's a current member of the Geology Foundation Advisory Council. Fred resides in Dallas, Texas, where he's the president of PVT Inc. In 2009, Fred's family established a lectureship, a scholarship to honor and recruit graduate students into the field of hydrology. Our speaker today will be introduced formally by Dr. Ashley Matheny, who is an assistant professor in the Jackson School of Geosciences. Ashley?
Thanks, Liz. It's my pleasure to introduce you guys today to Dr. Gaby Katul. So Gaby is currently a professor at Duke University, but he started out in Beirut, Lebanon, receiving his bachelor's degree. He transitioned to a master's degree from Oregon State and eventually completed his PhD at the University of California, Davis. So he is currently the Theodore S. Coyle Professor of Hydrology and Micrometeorology at Duke University. And he's won a very impressive list of awards, and I will only embarrass him with a few of them. That recently he's received the Hydrologic Science Award from the American Geophysical Union in 2012, the Dalton Medal from the European Geosciences Union in 2018, and the Award for Outstanding Achievements in Biometeorology from the American Meteorological Society in 2021, which is not quite happening yet. So Gaby's research and his lab group's research focus on micrometeorology like my own and near surface hydrology, with emphasis on fluxes of heat, momentum, CO2, water vapor, and all sorts of other things, including aerosols, pollen, and seeds. And he's really interested in water transport within the soil plant atmosphere continuum, and along those lines, the implications for that in hydrologic, ecologic, and atmospheric and climate change research. So with that, I will let Gaby take it away and tell us all about evapotranspiration from kinetic theory to the limits of plant life.
Thank you, Ashley. I'm going to go ahead and shut my video as well, so this way the internet will not be overcrowded on my end. Thank you all for having me. This talk is labeled as Evapotranspiration from Kinetic Theory to the Limits of Plant Life. This work is done in collaboration with my graduate student, Asad Murad, my new colleagues, Jean-Christophe Domecq, Sari Palmroth, and Ram Oren, my former PhD student, Shangwei Huan, who is now a postdoc at University of New Mexico, another former PhD student, Stefano Manzoni, who is now at University of Stockholm, my collaborator at Tübingen University, Wilfried Conrad, a former postdoc, Julia Vico, who is now a faculty member at the Swedish University of Agricultural Sciences, Sana Savento from Los Alamos National Lab, and Danny Way, who was a postdoc with Ram Oren at our school, and a faculty member at the University of Western Ontario. It may appear arbitrary to commence a general talk on evapotranspiration from the point of view of the kinetic theory of gases. At this point, ignores probably 1,000 years worth of inquiry into both evaporation and transpiration. And the reason for picking this starting point of this general talk is because the kinetic theory was the first to pinpoint the nature of the phase transition that is associated with evaporation. And it is also the mechanism or the statistical mechanism that has offered the first microscopic view of the phenomena. A brief introduction to the kinetic theory, the work started by Daniel Bernoulli in his book, Chapter 10 of Hydrodynamica, where the essence of the kinetic theory of gases was first established. I should say that the contemporary textbooks today still teach the kinetic theory of gases pretty much along the lines of that was outlined in Bernoulli's book in 1738. And interesting enough, as a bridge to current times, Daniel Bernoulli was also the first to investigate epidemiology from a mathematical perspective. It took almost 60 years before John Dalton realized the value of the kinetic theory of gases and was able to formulate the law of partial pressures to describe evaporation for the first time. And in fact, this was formulated as a resistance to vapor motion. He did not know at the time of how to craft this resistance per se, but he knew the driving force for evaporation, and this was linked to the partial pressures. It took another almost 60 years before the next phase of evolution of the kinetic theory of gases, and that came about from the work of Clausius. Rudolf Clausius realized that kinetic energy generalized to include translational, rotational, and vibrational components, and hence he was able to generalize Daniel Bernoulli's kinetic theory for gases to liquids and solids. And in it, Clausius was the first to introduce the concept of mean-free path. But 2 years later, it was Maxwell who was able to derive the statistical distribution of kinetic energy for gases, and that actually was a major breakthrough because it enabled others, including Ludwig Boltzmann in 1877, to make the connection between entropy and probability theory. And in it, in 1877, Boltzmann was able to unify basically classical laws of thermodynamics and statistical mechanics for the first time. And that was a major development for understanding what happens in closed systems, at least from the perspective of evaporation. To show that, in fact, one can look at a closed system, a box that is shown on the left-hand side, with molecules in the liquid phase, and at a given temperature, the molecules do have a distribution of kinetic energy. So there are some molecules that are highly energetic, others not. And what happens is that the molecules at the interface that are highly energetic will have a probability of escaping, partly because of collisions from other molecules beneath them or next to them that push them outside the liquid interface to the air interface in that box. And after a while, the number of molecules that can escape have escaped, and there's a certain equilibrium that is reached at this temperature, where the molecules that are escaping the liquid phase is roughly commensurate with the number of molecules that might hit back liquid-water interface. And when this equilibrium is reached, where the number of molecules escaping is balanced by the number of molecules that are recondensing, the evaporation rate is 0, and we call the air in the compartment of the non-liquid part of that compartment as saturated. Of course, if we increase the temperature, we increase the kinetic energy of the molecules that can escape. Those molecules then, there's more of them that can escape, and then after a while, more of them will recondense, and the new equilibrium is reached, where in fact, at a higher temperature, there will be more molecules in the air phase, and that means that the saturation vapor pressure, which reflects this equilibrium state when the evaporation rate is 0, is reached again. And one can see that as temperature goes up, the saturation vapor pressure goes up. It was quantified mechanistically by the Clausius and Clapeyron equation, and that basically sets the stage for what happens in a closed system. Unfortunately, hydrologic processes are not dealing with closed systems. They are dealing with open systems where both mass and energy can be exchanged. And so when this box is opened up, a whole slew of other processes become active. The first, of course, is the fact that suddenly these molecules are going to be moved around by turbulent eddies. And of course, in 1877, just the same year that Ludwig Boltzmann published his famous connection between statistical mechanics and thermodynamics, Valentine Boussinesq proposed the theory of the swirling and agitated flow of liquids. In fact, in 1877, I should say, the word turbulent was not known. So the analogy to it was that the flow was swirling and agitated, it meant turbulent. And the 1877 paper of Boussinesq introduced the concept of eddy viscosity for the first time. And this paper was transformative in several ways. Acknowledged the fact that turbulence must be analyzed in a statistical sense. And the concept of local averaging was first introduced way before actually Osborne Reynolds' work on pipe flow was represented. And the concept of now macroscopic mixing length, meaning mixing length of large scales, was first put forth by Boussinesq. That was the basis for the concept in fluid mechanics. And of course, this type of mixing length formulation at the macroscopic scale later was used as an inspiration by von Karman and Rind to derive the so-called log law for the viscosity profile. But this is all I'm going to say about aerodynamics at turbulent flows at this talk. This certainly warrants another seminar, maybe one day. But the focus here is going to be more on plants. And so just as a background for that, I wanted to make sure that these developments from a historical perspective are linked. Now, of course, the more important one to us is when we open that box, is the energy exchanges that we have to consider. And while those have been known for quite some time, the work of Ira Bowen at Caltech, in fact, perhaps considered to be the first major effort to put a whole slew of pieces together. And in it, Ira Bowen suggested that the energy arriving at the surface may be partitioned into sensible and latent heat fluxes. Latent heat fluxes is the amount of energy that goes into vaporizing the water. This was the first realization of how water vapor transport and evaporation rate from an energy perspective and the mass perspective were linked. Then Ira Bowen did something else. He used the Boussinesq approximation to link sensible heat flux to temperature gradient above the surface, and he linked the latent heat flux to water vapor concentration gradients above the surface using exactly what Boussinesq did, an viscosity or an diffusivity formulation. And then he realized that if the sensible heat flux to the latent heat flux are divided by each other, one can then define a ratio between these two fluxes simply based on the concentration differences of water vapor and temperature if the eddy diffusivity for heat and the eddy diffusivity for water vapor are the same. And in fact, Bowen made a mistake, so that he argued that if eddy diffusivity for heat and the eddy diffusivity for water vapor resemble their molecular counterpart, in other words, the ratio of eddy diffusivities in the atmosphere resembles that of molecular diffusion, and he argued that the Schmidt number in the atmosphere is nearly 1, then those numbers cancel, and therefore there is a certain similarity in eddy diffusivity for heat and water vapor simply due to the fact that the molecular counterparts also average out, which was erroneous, but got the right answer almost. And with that approximation, Bowen was able to infer the latent heat fluxes or the evaporation rate strictly based on the available energy and micrometeorological measurements that simply relate the water vapor concentration difference and the temperature concentration difference to heights. And it enabled, in fact, Ira Bowen to monitor continuously evaporation from lakes with that technique. After that, Howard Penman was able to revise Bowen's analysis by including more explicitly the aerodynamics, and that has led to what we now call combination equations for evaporation. It was a major breakthrough because for the first time, the role of aerodynamics and the role of energy were quantified. And at the same time, well, before that, but in the West, we knew about this formulation in 1954 by Monin and Abukov, the two Russian physicists working with the Russian mathematician Andrei Nikolovich Kolmogorov, derived the so-called similarity theory. This was theories derived in the 30s, but the American Geophysical Union in 1954 translated that word from Russian into English and basically knew about it at that time. And what was remarkable about this theory is that the evaporation rate was linked again to a water vapor concentration gradient above the surface, above the evaporating surface. But now the diffusivity that was speculated by Boussinesq was quantified as a function of the shear stress and an effective mixing length, as well as some adjustments due to thermal stratification in the atmosphere. So the first attempt to link the eddy diffusivity to a mixing length, as well as the role of thermal stratification that were basically expanding or contracting effective eddy sizes or the macroscopic mixing length that was transporting water vapor. So to summarize basically what the theory of evaporation came about was from the work of Dalton in 1802 that realized what the driving force for evaporation was, to Adolf Ficks's molecular diffusion representation that evaporation is simply a molecular process governed by a molecular diffusion, in the absence of turbulence, as we might expect in a closed compartment with turbulence, to the fact that when we open this box and we have turbulent eddies, the evaporation rate follows the same form, but now rather than deal with a molecular diffusion coefficient, we deal with a turbulent diffusion coefficient where the mixing length itself could vary with thermal stratification. I should say that this work was actually also proposed by Boudico. Boudico was a famous climatologist. He headed the same lab that Monine and Nabokov were working at. And in fact, his early papers in the 1920s were about the correction of the von Karman constant stability, because at the time he envisioned that it was the von Karman constant that was changing rather than the entire mixing length. But nonetheless, Boudico was able to formulate a model for evaporation, and later, of course, hydrology knows about Boudico through the famous Boudico curve. Up to this point, plants were invisible from this line of research. In 1965, John Monteith was able to revise the Penman equation to include stomates. And this was the first instance where a physical model suddenly had stomata in it. And basically, he added a more realistic formulation for the aerodynamic conductance. How to specify that stomatal conductance remained almost a mystery, but that was somewhat fixed by Paul Jarvis in 1976. Where he actually proposed a generic model for stomatal conductance, where stomatal conductance is some maximum value that occurs when all the stomates are open to the maximum aperture, adjusted by a bunch of reduction functions that might vary with photosynthetically active radiation or light, the vapor pressure deficit that we started the seminar with, the leaf pressure, and the atmospheric CO2. And I should say, when these formulations appear as such, when you have some maximum quantity adjusted by a reduction function, it means that the field is still in a stamp collection phase. There is still a data collection phase that is trying to be sorted out, where the key control variables on it are yet to be determined. And that was roughly the state in 1976, at least in the context of this type of model. And if there is interest in reading more about the history of evaporation, the book by Wilfried Broussard in 1982 has a full chapter. I think it was chapter 2, where the history of evaporation from pre-Greek era to the early 1980s was proposed. And in fact, Broussard says that this chapter was the hardest to write. Okay. In parallel to these developments, in the late 1960s to the late 1970s, computational power was sufficiently strong to begin to explore how climate models might sense land surface evaporation and transpiration. The role there was to figure out how the land surface impacts the climate. And of course, the focus was on transpiration and evaporation, as well as sensible heat. And there the focus again was those fluxes because of their role on precipitation. In other words, land surface fluxes impact boundary layer heights, and lifting condensation levels impact the necessary conditions for formation of rainfall. And this was a period where there was recognition that land surface processes could impact the climate system in ways that were perhaps not appreciated as much. And because of these sensitivity studies from the climate model's perspective, the first basically large-scale field experiment began in 1986 in southern France. This was the first attempt to instrument a field site at the grid scale of a general circulation model. And it was one of those experiments where there was a lot of coordination between radio soundings, land surface measurements, leaf level measurements, aircraft measurements, satellite remote sensing measurements. Lots of remote sensing products came out of that experiment. And so basically the era of large-scale field experiments and their success began from this 1986 experiment in southern France, and many of them later on were repeated in different regions around the world. And in fact, the recognition of remote sensing products became quite evident after the experiment. But up to that point, the CO2 story was somewhat implicit. It was not really in the core of these experiments. If one looks at the HOPEX mobility data sets, there was few mention of CO2, but very little. And the story of CO2 is no less exciting than the story of water vapor. It started by Jan Baptist van Helmholtz in the 1600s, who actually was the first to coin the word gas. And he also noted that the gas silvestre, carbon dioxide, is in fact given off by burning charcoal. Also, Van Helmont was the first to investigate actually the conversion of water to carbon through his studies of gas exchange from willow tree. And in fact, what he found is that if you plant a willow tree and you keep watering it, after 5 years or so, that tree grows. And what Van Helmont did is he weighed the soil, found no major difference. He weighed the plant and found a lot of difference between the original weight and the final weight, and came to the conclusion that the addition of water somehow increased basically the carbon weight of the plant. So the link between CO2 uptake and water, so to speak, water release, because he was adding water to keep the pot irrigated, became quite evident to him. And that was basically our first inkling that, yes, there is a connection between CO2 uptake by vegetation, water release to the atmosphere, and basically plant growth. So this was all done through the work of Helmont. Interesting enough, centuries later, one can say that the activities of Van Helmont converged into almost a modern day story. Atmospheric CO2 is rising largely because of fossil fuel combustion, whereas the ability of plants to uptake CO2 is perhaps one of the leading mitigation strategies to offset this rise. If one looks at the current estimates of the global carbon budget, one sees that the land sink is perhaps one of the dominant sinks in the system. And so clearly, whatever we do to that sink is going to have a huge impact on the buildup of atmospheric CO2 in the atmosphere. And this clearly became a focal point, basically, to climate models. So one can say that from the mid-1990s onwards, general circulation models witnessed almost a renaissance in terms of adding physiology and biology to them. The greening of GCMs basically was happening at that era, and several influential papers about the carbon cycle and the water cycle appeared, where the effects of elevated atmospheric CO2 on the ability of the biosphere to suck up CO2 from the atmosphere became more evident. One of the leading papers appeared by Peter Cox and co-workers in 2000, where they actually showed that if CO2 keeps going up, stomatal conductance will drop. And because of that drop, there is less and less uptake, and that might be the acceleration of global warming. And there were other corollary issues about the water cycle. So, for example, Betts and Gedney and others have found that because of increased atmospheric CO2, stomates open less, so more and more of the precipitation patterns will end up in increasing continental scale runoff. And so basically the connection between stomates that are really operating on less than a micrometer scale to the climate system began to be realized in these studies. Now, of course, at the same time, long-term experiments were also being conducted. These are forest growth and forest inventory experiments that were happening all around the world. And there was some recognition that droughts were beginning to amplify, and the forest mortality was occurring more frequently at these long-term experiments. And so that actually shifted a little bit the view from how stomates impact the climate to how the climate now impacts plants. And that is going to be the focus of this talk. So to do that, of course, we're not going to be satisfied with a description that is strictly limited to the Penman-Monteith equation. We need a little bit of a more sophisticated machinery to do that. And for that, one needs to understand the plumbing within the plant system itself. And this has been going on for some time. We've mentioned the work of Adolf Fix in 1855 regarding gas diffusion. So if one starts at the leaf level, it has been recognized that when stomates open, CO2 enters, water vapor leaves. And so basically, CO2 enters to satisfy the biochemical demand by the plant, and the penalty that the plant pays is that water molecules escape. The plants actually, stomates themselves are replenished by liquid water from the xylem. The xylem is basically a plumbing system that is pulling water, that is allowing water to be transported from the soil all the way to the leaf under tension, which means that it's in a metastable state and therefore susceptible to cavitation. And of course, the products of photosynthesis, the reason the plants photosynthesize is to use carbohydrates and sugars so that it can produce seeds, it can grow its roots, it can replace its foliage, it can increase its biomass, and so forth. And it turns out that the sugar transport, which are the products of photosynthesis, occurs in the phloem, and that's primarily happening through osmosis and osmoregulation. Sugars pull water into the phloem system, thereby pressurizing the water in the phloem and pushing sugars and water down to where the sugars are needed. And so while water is moving from the soil to the leaf under tension, sugars and water are moving from the leaf back to the soil under positive pressure. And so this is the basic plumbing machinery in the plant. In each compartment, whether it be the leaf, the xylem, the phloem, and of course the soil, there are a number of theories that have been proposed. And we're going to go through them, but the key ones is that in the case of the xylem, the so-called cohesion tension theory has gained prominence in describing water movement in the plant. And the phloem, of course, the monk hypothesis or the osmoregulation hypothesis gained some prominence in how sugars are then moved from the leaf all the way down to the sink. The phloem will not be the focus of this talk because we're going to be focused primarily on xylem ecosystems, but I mention it because it is actually tightly coupled to what's happening in the leaf and at some level what's happening in the xylem. In each one of those compartments, a number of theories have been proposed because, as we shall see in a minute, the number of unknowns are more than the number of equations. So to close the problems mathematically for models, one has to make extra assumptions or conjectures or hypotheses as to how the plant operates. And in the case of the leaf, it has often been viewed that stomates operate in a certain way. The idea is that stomates are somehow optimizing the passage of assimilates while preventing excessive transpiration. These ideas that stomates are somehow operating optimally to maximize the carbon gain while minimizing water losses was put forth by Cowan and Troughton in 1971, later by Boussinesq and others in 1976, still going on today. In the case of the xylem, the description of the xylem, while the physics of it are beginning to be understood, why the xylem is structured in a certain way, what are the topological feature of this xylem remained a little bit of an unknown. And so certain hypotheses have been framed about the fact that the xylem may have evolved in a way to buffer the plant against variability in the environment, which led to the so-called safety efficiency trade-off. This is basically a sort of a trade-off where the plant has to remain safe because there are some surprises that are going to be encountered during the many years that trees usually live, versus the need to allow water to move efficiently from the soil to the leaf. And so that led to the trade-off itself, that if you try to build vessels that are too big and efficient, then they might cavitate quite rapidly. Whereas if you build vessels that are too narrow, you actually minimize the possibility of cavitation, but you lose hydraulic efficiency. And the same thing can be said about the phloem. Basically, there are two competing forces in the phloem. If you increase the sugar concentration in the phloem a lot, you increase the osmotic pressure that drives the water, but at the same time, you increase the viscosity of the fluid by increasing the sugar concentration, and that has an opposing effect. So there is clearly, again, in the case of the phloem, some optimum concentration that allows efficient sugar transport, where there is some compromise between the need to generate an increased pressure gradient for osmosis, but because of viscous buildup with increased sugars, there is some resistance to that. I mean, think of maple syrup, very viscous, yeah? Okay, so just to show now basically how the leaf level processes are operating, and from a mathematical point of view, what are really the issues and the problems, one can start by describing the biochemical demand for atmospheric CO2 by the plant. And that is often encoded in the so-called Farquhar photosynthesis model that links the rate to a state, the rate here being the uptake of CO2 by the plant or the biochemical demand for CO2 by the plant, and the state is the internal CO2 concentration in the cells of the leaf. And that is basically the saturation function. As the internal CO2 concentration increases, the biochemical demand goes up to a certain point, and then basically almost saturation. So this is a mathematical form that links a flux to a state, and the relation between the two has three parameters labeled here as alpha 1 and alpha 2 and gamma star. Alpha 1 and alpha 2 are basically a function of light and temperature They are determinants of whether photosynthesis is sometimes limited by the Rubisco enzyme or by other processes such as RUBP regeneration mechanisms, which are light-driven. And so the photosynthetic rates mathematically, however, is still encoded in this expression, where alpha-1 and alpha-2 can vary with light and temperature. Now, this demand is being supplied by the atmosphere. So if one again links the flux of CO2 to the internal CO2 concentration gradient by knowing the atmospheric CO2 concentration around it, one sees, again, a link between a flux and the state. And even if we know the CO2 concentration around the leaves, and that tells us how frequent CO2 molecules are bombarding the leaf and therefore able to enter the stomates, the aperture of the stomates, how many of them and how open they are, is unknown. So while we've added one equation that allows us to link the supply to the demand by equating, that's a steady state argument, we are left with two equations and three unknowns: the photosynthetic rate, the stomatal conductance, and the internal CO2 concentration. And there lies the problem, that physics and biochemistry alone, even in the most simplified case, are insufficient to allow us to describe how photosynthesis proceeds. So we need this extra hypothesis about how stomates open and close. And what has been done in the climate models is to simply assume some sort of an empirical link between stomatal conductance and photosynthesis, often normalized by atmospheric CO2 concentration, adjusted by some atmospheric aridity factors such as relative aridity. And while there are numerous formulations like those around, they all share a common finding that the relation between stomatal conductance and photosynthesis normalized by atmospheric CO2 is sort of linked. And so that was kind of a key finding. These are the same models that started making their way into the mid-1990s in climate systems, where the greening of the climate models began in fact using these two formulations. But the more important thing for us is that there is a clean relation between stomatal conductance, photosynthesis, and atmospheric CO2 that we will try to recover in a slightly different way. And that different way revisits the approach by Cowan and Farquhar in 1977, where rather than ad hoc formulations for linking photosynthesis to stomatal conductance, one might revisit the idea of optimality. And there is a whole mathematical machinery that can be thrown at it. So if we envision that the uptake of CO2 is a carbon gain for the plant, so that's a gain. Water losses are a cost to the plant. They are still being driven by the same pathway, stomatal conductance. Now we can go ahead and decide, well, how should the plants open and close its stomates to maximize the carbon gain, subject to a constraint that the water is available in the rooting zone. And to do that, to solve these types of constraint optimizations are quite difficult. The mathematical trick is to convert these constraint optimizations to unconstrained optimizations of the Hamiltonian and Lagrange multiplier. So a Hamiltonian can be formulated based on gain minus the cost via a so-called Lagrange multiplier. The Lagrange multiplier ecologically means cost of losing water in carbon units. So that is kind of a bridge that links the carbon economy and the water economy together. And under those conditions, once the Hamiltonian is formulated, it's a function of control variable. The control variable here is, of course, stomatal conductance, GS. That's how stomates open and close. We're ready to solve the so-called Euler-Lagrange formula. In this case, the Hamiltonian is not sensitive to the velocity of how stomates open and close. So that goes to zero, and we are left with the finding that the rate of change of the Hamiltonian with respect to the stomatal conductance must be zero. And that means that if we are able to express photosynthesis as a function of stomatal conductance and water vapor as a function of stomatal conductance, we are able to solve at least this primitive Euler-Lagrange formulation problem. And that can be done if we see that we can link the biochemical demand to internal CO2 concentration. We have the atmospheric supply linked to CO2 concentration. So we can, in fact, get rid of the internal CO2 concentration and write basically the photosynthesis function of stomatal conductance. Using Dalton's law, we can write the water vapor flux as a function of stomatal conductance. And basically, we impose as a third equation now the optimality hypothesis that the rate of change of the Hamiltonian with respect to the stomatal conductance must be zero. That makes a certain assumption about the constancy of the Lagrange multiplier. and how to determine it. But mathematically now the problem is closed via an optimality hypothesis. In other words, we are assuming that the plants are operating in a certain manner to maximize carbon gain for a given amount of water in the rooting zone per unit leaf area. In principle, the Lagrange multiplier can be determined by the constraint that the water is available in the rooting zone, and hence the problem for all practical purposes is mathematically closed, and it can be solved for all types of limitations on photosynthesis. We've done that in one of the papers, and I simply would like to show some intuitive results that pop up from this type of approach. So, for example, if photosynthesis is linearly related to internal CO2 concentration, that means that we are looking at a linearized biochemical demand function, which would be more appropriate for visco limitations of photosynthesis. One can then differentiate the Hamiltonian stomatal conductance, set it to 0, and solve for the optimal stomatal conductance as a function of the slope of the photosynthesis internal CO2 concentration. And those results are shown in the slide. They predict the stomatal conductance as a function of CO2. They predict internal CO2 concentration as a function of CO2, and they predict photosynthesis as a function of CO2. And one thing that could be interesting to point out is that if you divide photosynthesis by stomatal conductance, that slope cancels, and one can recover, in fact, the empirical relation between stomatal conductance, photosynthesis, and atmospheric CO2. And moreover, one can predict the atmospheric aridity to be vapor pressure deficit to the -1/2. This is coming from stomatal optimization theory. It's an emergent property of the stomatal optimization. And basically what it says is that stomatal conductance does vary linearly with photosynthesis and atmospheric CO2. The Lagrange multiplier at the cost of losing water and carbon units becomes obvious. If you use more water than the plants can afford, this cost goes up, so stomatal conductance goes down, and so forth. So a complete picture is beginning to emerge, where at least this exponent -1/2 is an emergent property of the optimization rather than externally imposed like relativity or pressure deficit. And this result is robust. It turns out that if you repeat the same analysis for other limitations, such as the RUP regeneration mechanism, as Belinda Medlin did in 2011, you get a form that is quite similar. How well these models work, basically, if you look at data sets that are coming from gas exchange, as shown in this picture here, and you plot evaporation from these gas exchange measurements as a function of the square root of vapor pressure deficit times photosynthesis, you get a straight line. These are data sets coming from Scott Spine that was collected across a climatic gradient in Europe, published by my colleague, Sari Pamroth. And again, the agreements are quite consistent with stomatal optimality. Okay, so this has been pretty primitive in terms of stomatal optimality theories. There are other ways to complicate the story where the amount of water now is fluctuating, and that moves us into a domain that is called dynamic optimality principles. Asad Murad recently did that with Sanna and JC Tomek and Yan Lan and Mazen and myself. And basically, you could show that you could now write a dynamic optimality relation where the Lagrange multiplier itself evolves in time, depending on the water strategies, and things such as isohydric or anisohydric of plants from this approach. I will not delve into the details other than to say that if the plants did read the mathematical textbooks, more important, they understood it, then one can understand why they're behaving a certain way. Okay, so now moving from the leaf to the xylem, which is our next focus. Now we've talked a lot about how water is lost from the leaves and how CO2 enters. What we would like to talk about now is the delivery of water from the soil all the way to the leaf. And as we have said before, this is occurring through water conducting cells. And the idea is that as water molecules escape from the leaf, for every water molecule that you lose, you have to pull an entire column of water all the way up by one molecule. And that is what is increasing the tension, so to speak, in the xylem. For every water molecule you lose, the water column from the soil all the way to the leaf has to be pulled up by one molecular diameter. So of course, if you lose more water, you have to pull that column more and more. And that means that you are relying on the adhesive properties of water to keep that column intact. And this actually view, which is the essence of cohesion tension theory, was pretty debated. In fact, it was a very contentious debate in the 1800s between two big camps. These camps were labeled as the physicists on one end and the vitalists on the other. The vitalists were arguing that living cells should play a role in the transport mechanism of water, but at that time they did not know how it happened. They could not identify which cells are really active in that transport and what are the details of that transport mechanisms. And this debate continued until the experiments by Edward Strasburger in 1891 confirmed the physicist's view. And what he did, in fact, was somewhat interesting. He immersed the roots of a tree in copper sulfate. So that killed all living cells, basically. And what he found is that water is being transported in the xylem for 3 weeks or 4 weeks after all the cells have died. And so that actually confirmed the fact that the vitalists were not correct, but not proved were completely correct. Based on these results, a major advance happened by Dickson and Joly, where they used the insights from the prior experiments to delve deeper into what was happening in the parenchyma. And they found that there was a curved meniscus at the interface that develops within the leaf parenchyma. And that's exactly where transformation was happening. So they said, oh, well, a curved meniscus, that means surface tension is able to pull carry the water column itself and even pull it against gravity and overcome the frictional resistance when water molecules are being lost. And of course, this was again highly contentious by Francis Darwin, the son of Charles Darwin, who was kind of the leading biologist at the time in the UK. He criticized the Irish duo Dickson and Joly for being completely off, accusing them that the ability of water to withstand tension is some of us equivalent to believing in ropes of science. That's how he labeled cohesion tension theory. Unknown to Darwin at the time was the experiments by Berthelot in 1850 and Jaeger in 1892 that demonstrated that, in fact, water can withstand both compression and tension forces, but not shear forces. And that's where the confusion of Francis Darwin happened. He envisioned tension forces as the equivalent of shear. And of course, fluids cannot withstand shear, but they can certainly withstand tension. And the experiments of Berthelot and Jaeger actually showed that water has a huge capacity to withstand tension. In fact, forces as high as 100 megapascals are needed to break the hydrogen bonds, even though the experiments at that time were controversial because some thought that this was not measuring the cohesive forces of water molecules, but the adhesive forces of water on solid tubes. However, the fact that such high pressures such high tensions were withstood by the water column proved that the cohesion tension theory may have its own merits. But it took actually a little bit longer to understand what was really happening, though. The view that was taken by Dickson and Joly is that cavitation was somewhat a rare phenomenon. And it took actually a little bit of time to understand that this was not the case. Primarily that came from the forestry work of Haberland, who in 1914 actually stated that vessels and tracheids normally contain both air and water. So the presence of air in the tracheids was not a surprise, of which he argued that the relative amounts of the two substances could vary according to the season and even the time of day. So he already knew that there were some diurnal variations in the amount of air in cavitation that may have happened in the xylem system. And in fact, his view was again controversial. It was rejected initially, but later confirmed by Milburn through wonderful experiments where acoustic measurements detected, in fact, the formation of gas emboli, and was able to show that even during reasonably well-watered conditions, cavitation may, in fact, still be happening in the xylem plants. Okay, so now that we have established that cavitation can occur, how do we quantify its impact, and what does that have to do with plant responses to stress? That's a basic problem in ecohydrology, and we know now quite a bit about how plants respond to it, and that response is often quantified by the percent loss in xylem conductivity, or PLC. And this picture shows the percent loss in xylem conductance on the y-axis against xylem pressure. This is negative. On the x-axis, then you could see that riparian trees or desert shrubs have very different responses. Riparian trees, of course, you apply a little bit of negative pressure and you lose conductivity of the xylem pretty rapidly, whereas desert shrubs, you could apply xylem water tension up to 8 megapascals and still you hardly lose 50% of conductivity. Now, of course, this is a macroscopic quantity that can be used, but the question is, how can we derive it a priori for species where we cannot conduct those measurements? Of course, one way to do that is to try to measure it for certain groups and assume that these groups function the same way. The other one is perhaps to borrow from ideas from soil physics and ask very much like what people ask in soil physics. If I know the pore structure of the soil, can I predict its hydraulic conductivity or gas diffusion or electrical conductivity or thermal conductivity? And basically, that's what we will try to answer a little bit next. Can wood anatomical traits give us a clue of how to describe the hydraulic conductivity or the percent loss of conductivity of the xylem? And the idea is as follows. There are clearly different shapes. One looks at, for example, a ring porous wood structure. So this is a graph that shows vulnerability curve, xylem pressure on the x-axis, the percent loss in conductivity on the y-axis. For a species that is ring porous, its wood actually has big rings early on in the growing season, and as the growing season progresses, the pores become smaller and smaller, and then the next season comes and the pores are bigger. So again, you see them here, but then as the growing season progresses, they become smaller. And often than not, if you look at the vulnerability curve of these types of species, like what we call an R shape. On the other hand, if you have a diffuse porous system, the choice of language here is obvious. The pores are all diffused from year to year. So 1 year, this is another year. You find that, in fact, there is more uniformity in the pore sizes. There are no big vessels like you see in the ring porous. And basically, the vulnerability curve looks quite different. And of course, one can see that this is quite efficient at transporting water, but it's not very safe. So it's competitive when water is available. Whereas diffuse porous, it's quite competitive when water is scarce because it can handle high xylem pressures. And the question is, can we derive those curves by knowing something about the wood anatomy? And to answer that, one needs to dig now deeper in how water flows in the xylem system. And basically, if you look at the anatomy of the xylem system, you find all sorts of scales, anywhere from centimeters to nanometers operating in water transport. At the centimeter scale, you could see that the xylem itself that is pulling water from the soil over the leaf is composed of tracheids or vessel members. Those are of the order of centimeters in length. Their diameters are in micrometers, if you like, and water moves up to this point, and then it has to cross a certain membrane. That membrane is composed of many pit fields, and within each pit fields, there are very tiny pores that are of the order of nanometers. So to model water movement, one has to be concerned with the axial flow and the radial flow across those membranes. The axial flows through the vessel membrane. And to do that, we tried to build a model where initially the representation of the pores is a porous, diffuse porous structure. And we said, well, okay, this is a good starting point. Yeah, it's kind of symmetric, so 2D representation might be good enough to do these calculations. You see here the kind of pore structure that you see, and those actually are cavitated vessels themselves. The model is built as such. You preserve the statistical properties of the allometry, the length and the diameter, and the number of contacts that long tubes will have with other tubes. And basically what you do is you then allow them to connect through membranes where the pore structure of the membrane is prescribed. The statistics of the pores are prescribed. It's assumed also that all vessels are vertical, and so all the vertical movement is going to be happening through tubes, and then all the radial movements is going to be happening, or at least encountering resistances to the pit membrane, and hence it's going to be a very different of which energy is being lost when you move water through this membrane. Cavitation is assumed to occur when the largest pore is heated, so air can enter depending on how big those pores are. And of course, there are other competing hypotheses, such as membrane stretching, but membrane stretching simply increases that pore to the leading order. the physics behind it is that the Hagen-Poiseuille equation is used to describe basically the flow in the tube, the axial flow. And in this case, the pressure drop is inversely related to the force power of the radius of the tube, whereas the radial flow or horizontal flow is actually not given by the Hagen-Poiseuille equation. And that's what makes actually water flow in the xylem system very different from soils, is that there's different physics involved in the two directions, the axial and the radial. That is actually the radial part is given by a Sampson. This is actually the pressure drop that you see if you have a sheet, a very thin sheet with a perforation. The pressure drops from that to force water to flow is in fact inversely related to the cube of the radius of the pore. So very different physics involved in the pressure drop across the vertical direction and the radial directions. Putting all of this together and running a model that is basically loaded with the structural properties of acer glabrum with a forest species, one finds that as you start increasing the pressure of the air, so this is how we are increasing potential for cavitation, you see more cavitated vessels in blue. You see isolated vessels. So these are vessels that are still carrying water, but they become hydraulically disconnected from the remaining network. And of course, there is still some functional vessels that are left. And you could see that as the pressure of the air, which is equivalent to increasing the tension of the water, goes up, there is more and more cavitation. How does this model reproduces the hydraulic properties of acer glabrum? Well, the percent loss in conductivity reasonably reproduced by these calculations when all wood anatomy are loaded. Other things that can be predicted from this model is the relative conduit water content. Because of isolated vessels, these are vessels that become hydraulically disconnected from the remaining hydraulic network, but they still carry water. So in fact, while hydraulic conductivity is lost almost 90%, as the pressure increases, the relative conduit water content can be as high as 50%. And that might play an important role in the recovery from embolism as the drought eases or as more water becomes available in the soil. These calculations are done using a network model that is freely available on GitHub if you want to download it and play with it and upload different species. It's described in that paper. And basically, we are at a phase where now we are ready to make some conclusions. So the specific conclusions from this work is that it is possible to predict the shape of the vulnerability curve from, and as well, relative conduit water content from wood anatomical traits. There are multiple scales involved in describing xylem flow, anywhere from big, centimeters, this is the vessel length, intermediate, which is the vessel width, to nanometer scales, which is the tip diameter. And the shape of the vulnerability curve is impacted by all of them. We should also say that this is exciting time for ecohydrology because there are now multiple techniques, X-ray tomography, magnetic resonance imaging, and so forth, that are able to image now the wood anatomical structure, which we never had before at this resolution. So this offers a new perspective on how to quantify vulnerability to cavitation impacts. And last, I should say that problems of networks are, of course, very common in many branches of physics. There is always this debate of what happens whether the components of that network are operating autonomously or are they part of a more coordinated network. The coordination here means that are the hydraulic properties of the plant out of whack, so to speak, with the photosynthetic properties, out of whack with the transport properties and the phloem, or are they coordinated? In other words, there should not be in this network a single bottleneck that then impedes the transport in the entire network. And that is opening up a new field in physics called networks on networks. And it is allowing physicists to explore issues such as why natural networks that co-evolve, species are co-evolving with the variability in nature, seem to be more resilient than man-made networks, say like the telephone lines or the electric network or the internet or the electricity grid or so forth. There seems to be more resilience in nature. The other thing that maybe this work can benefit from, again, making contact with a more contemporary topic, and I'm happy to take tons of questions on that last point if there is interest, is disease spread. At the end of the day, cavitation spread in plants is almost analogous to virus spread in human populations. Perhaps how plants have generated certain resilience cavitation spread might teach us how to think about perhaps containment strategies when we are encountering infectious disease spread in human populations. And I'm happy again to take more questions on this point if there is. Thank you very much for your time.
All right, so I think we've reached our time, unfortunately. So thank you so much for the great talk. I loved what you said about models. It applies also to tectonics, in fact.
Well, everything that has high dimensional
Yeah.
problems have the same issue.
Yep. Okay, but we should probably end it here. So please, everyone, come back next Thursday for our next DeFord lecture, and I'll stop the meeting now. Thank you so much. Thanks, Ashley, as well.
Thank you for having me again, Ashley. Thanks for inviting me.
Bye-bye.